thank you so much. So my talk is going to be a bit of a mix of things. I want to first talk a bit about my journey into a more support role in microscopy. I want to also talk about my facility, and then I will explain how a facility works, because I think not everybody is aware how we actually work. I will move over into the big elephant in the room, the data analysis, and then I would like to finish just with one of our communities here in UCL called BIG. So my journey into microscopy support role. So how I became a microscopist, I have this question quite a lot of times, so people think that we just pop up here next to a microscope and that's it. So as Joanna mentioned, I did my PhD in Spain quite a long time ago in cell division and PIC kinase. I'm not going to go into details about the science because everything, luckily enough, is published. But it was a very microscopy-based project that I enjoy a lot. I enjoy it so much that I moved on into the UK to carry on and doing a postdoc in mitosis again, this time studying kernel 1. And again, it was a really heavy microscopy-based kind of project. And again, it's published, so I'm not going to go into the details. I can ask questions. I mean, I can answer questions if you have anything about that, but I think it's very well published, and that's it. So postdoc is being a postdoc is great, I think. You do a lot of things, you meet a lot of people, you do a lot of experiments, and you have a lot of responsibility to publish, to look for the next job, to maybe thinking about writing grants, and so on. So at one point in the postdoc, I'd say, well, what I want to be in the future. So everybody talks about either academy or industry, but I wasn't sure what are the choices if you don't want to become a PI, what you do in academia. And I don't really, at that point, I didn't really know how industry works, especially in a country that is not my country. So the only thing I knew is that during my research, I really, really like the microscopy part. So I just saw a job looking for a microscopy support role in a core facility, and I thought, well, I do really like microscopy, and you can never have too many microscopes. So this is a screenshot of our light microscopy video on YouTube that explains light microscopy very, very well. So I decided to become a microscopy technician, confocal technician. We have many names, and don't listen to the other person. You never have too many microscopes. You can always have space for a new one, especially if it's fancy and new. So I want to put here this for everybody to see, because I don't know if everybody is aware of the technician commitment. So right now I changed from a research pathway to a support role, in this case, the technician pathway. The technician commitment is a framework that works to work in these 4 key areas: visibility of the technician, recognition, career development, and sustainability. I think, let me see if I can actually do the laser point. So visibility and this are pretty obvious, but career development in technician jobs is kind of lagging, especially at least what I'm aware here in the UK. So I really think that it's good for every technician working as a technician to be aware of this thing and push for change in the higher education. So after that, I'm going to do an overview of my place where I work. Right now, I'm currently working in London, as Joanna says, in University College. My core facility's name is the Center for Cell and Molecular Dynamics. This is the typical image of London, but we're working here in the Rockefeller building, very near to Houston. If you want to come and use our facility, you are always welcome. So more or less, we have around 17 microscopes. Hopefully, we will have 2 more soon, because you can never have enough microscopes. We also have 2 CT scanners. That's quite of rare in a light microscopy facility, but they are very well used, and it's something historical for our core facility. We also have 4 powerful computers, and the most important thing is that we are quite active. We have around 200 active users across 30 groups, and we are used not only for people from UCL, but we are open for people outside UCL. So the techniques that we work is the things that you see here. I didn't want to bore you to death with all the microscopes that we have, but we have everything from wide field to confocal to super resolution, to light seed, to high throughput, and as I mentioned before, we have also X-ray. And this is just a QR code if you want to see our facility. Because I actually didn't want to go much into the details. We have a website where you can actually see all our confocals. I actually did that website, so I'm quite happy with how it looks like. And you can click and pick and choose which one you are more interested on. And hopefully at one point I will move to click something. And so yes, so for example, just by chance the spark, you will see here all the specs of our system and a fancy image that I pick up to just impress people, and because I really like microscopy. So can I remove this? And then this is a lysine microscope that I think is really cool too. And the way the core facility operates is like, just in case people are not aware how to use a core facility, we will have a rate depending on where you are based, and it's supposed to be track charge. You will have a training, and then our goal is that you are able to use the microscope by yourself. That's the end goal. And all is very nice. We have a lot of microscope, but I want to make the point that all these microscope will mean nothing if there is no people that know how to use them and support the research that is done with those microscopes. This is just a very busy slide, just to show you that we are a healthy team of people. We all have different expertise. We all know how to use all the systems, but we will have our own expertise. And that's what I think makes the core facility works, the combination of the cool microscope and cool people. So how we support research, how a microscopy core facility support research. So we are from everything for experiment planning to training to use the microscope to help you during the acquisition, or even do the acquisition from you if that's something that you need. We offer data analysis help. I will talk more about that later. And here I put help collaboration and a lot of, because we do everything under the sun. I mean, whatever you need from with your science, with a microscope, we will try to make it happen. And talking about how a microscopy experiment works, the way people think is that you will prepare your sample, and then you will go to the microscope, you will take the image, and then you will analyze the data. And I will be very nutty, because this is actually the proper cycle of a experiment for microscopy. As you see, it's a cycle, because everything will feed back in this direction, but also in this other direction. In the past, people will prepare the sample without talking to the people on the microscope, so the sample will not be the appropriate sample for the microscope that they are available to use. And then they will acquire the image, and then when they are going back to analyze the data, the image, even though they are absolutely perfect, they are just not the image that you need to answer the biological question that you're asking here. That's why you always need to go back and feedback every step back and forward. So I leave this paper because I really like a lot this paper, and I send it to the people I train. It's a guide to planning and performing your experiment, and it has this figure that is quite useful to understand. So, for example, if you just want to see if your drug is doing something with the cell proliferation, you can actually just plate it in a multi-well, maybe use a high-through microscope, and that will give you enough data to answer your question: is my drug actually impairing the division of these cells? But if you want to do something a bit more let's say, a bit more complicated, seeing if your protein change localization, even though here they say Y-field, normally people will do that in a confocal just to make sure that the location of the protein is well defined in the confocal plane. So you will need to prepare the samples in a different way, and then you will analyze them in a different way. The important thing is that whatever you acquire is able to answer the question that you have at the beginning. And this microscope is no worse than the other one, and so on. It's just every microscope is just good enough if it gives you the data that you need. So the way we support research, as I said, this is just an example of one of the things I've been doing, particularly myself. This is a group from Imperial, and I've been involved in the optimization of the protocols, because it's when you see the final image in a paper, it takes quite a while to optimize the fixation, the permeabilization, the antibody labeling. And now we are in the processing of acquiring and then feeding this acquisition into the data analysis. And as I explained before, even though these images look absolutely perfect, they are very difficult to quantify. So maybe even we need to think about changing the protocol again. It's something that can happen. And as I mentioned before, we also support data analysis in the sense of help so far. So this is just an example of another, this is a lab here in UCL, Anna Frank lab, that works with pupa from Drosophila, and these are fat body cells, and I've been helping her to try to segment the cells. That is something quite difficult from the point of view of microscopy. So that leads us to the data analysis. That is something I'm very passionate right now, and I want to talk a bit more. So this is a very funny video. This is me, that I have plenty of experience of using Fiji or ImageJ, whatever you call it. And this is me trying to find a way to segment these cells. First, I will apply a Gaussian just to remove the holes, and we will be able to segment just one nuclei at a time. And as you see, even though I've been using this program for years, I don't remember where the buttons are, so that's why this is real time me playing with Fiji. So don't feel bad if you also take 20 minutes to find the threshold, because we all do that, and then I don't remember if I need to apply or not. And so this is real time. But my point of this video is like, okay, I'm doing this. This is just one just one image. Imagine if I have to do this with 200 images from my experiment, from my high throughput experiment. It will be very time consuming. It will be biased in the sense that I will be doing better maybe in the morning because I'm more fresh, and in the afternoon I lose the will to live, and I will make mistakes. So there is an inherent bias. So the next thing to do is to actually have a macro in Fiji. That will be like the next step of microscopy. So you have written a macro, you will apply the same macro to all your images. I also struggle to find my folder, but you see it's exactly the same result, and it's faster. But what will be better than just a macro in Fiji? This is a screenshot from GitHub repository from the Francis Crick Institute, from a course they do that is called Introduction to Image Analysis. The QR code is here if you're interested in doing that. This course is really good. So why this is better than my macro? Well, in here everything is explained and annotated. It's very easy to use. You can share it very quickly. Maybe it's even easy to be used for people that have no idea about coding, and because it's Python, you can actually start. If you see this, this is me running the same experiment with this script in the same data. You can start plotting some data and visualize your results just in the same script. You don't need to change to Excel or GraphPath or whatever you use to do that. So it's already more efficient. It's already giving you more data, and it's easy to share. That is something very important that I will talk later. So it's something that we are getting our core facility not there, but right now we have a new person in the division of bioscience that is called, his name is James Gilbert. He's a senior research data scientist, and he's also very passionate in this kind of being more efficient with the data analysis. So this is the whole idea for the future. We will have a proper data management, we will have data analysis, and we will go to data publishing. I not go into these two things in my talk. And our idea, and the idea of James, is try to transition into those more efficient ways of analysis to help researchers to do their research faster. That actually is the goal of a core facility. So our idea, the idea of James, is to make reproducible workflows with scripts that you don't need to know coding to be able to use them. That's really important. And we will validate everything with a test data set, and we will be able to run those pipelines in our high-performance computers in UCL. So for that, to start this project in the core facility, James asked for this grant, also the QR code here. It's from the UK Institute for Technical Skills and Strategy. And we were very lucky to have this summer intern, Lada, during 6 weeks. I can't remember if it was 6 or 8. She worked so hard. It looks like she was here for much, much longer. to start this project in the core facility. So her goal was to actually create a pipeline to do 3D segmentation, especially, even though here they say everything, she focused in 3D segmentation. That is one of the most challenge that we have here in the core facility. Normally people work with maximal intensity projection or 2D data. And she managed to create a very nice pipeline that is is working already and is just ready just for the last step to be able to be used in the high performance computers, and we will need to do some validation. But just to show you some brief examples that the things that she did in the summer, she first the pipeline will first segment the nuclei, then it will segment the cytoplasm, and then you will have different functions to segment things inside the cytoplasm. Depending on your organelle, you will have different functions to run, and you end with a table with all the data there. So it's very, very complete program. And I imagine that there are people here wondering why I'm being so annoying talking about Python scripts when there is programs that you just put your data there and it's really easy to click, 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 and you have your data. The first thing is that I never have access to those kind of programs during my PhD. They are very expensive, and not everybody has access to that. That means that if you want to share your pipeline with other people, and when you publish, you want people to be able to use your pipeline of analysis. So you will not be able to do that, even if you create a batch analysis program in this analysis commercial software. If they don't have the same version that you have, it will not work. So there is that limitation of people not being able to access those programs. Also, if you have the program right now, maybe in your next step, you will not have it. So I think it's a really good investment to try to learn open source of analysis software. So, and that brings me to, I copy this from the Euro-BioImaging website. When we talk about FAIR in data management and in data analysis, we talk about this as an acronym. So the data needs to be able to be fine, it needs to be accessible, it needs to be able to be interoperable, or whatever you say that in English, and we want to reduce the data as much as possible. And I'm showing you here in the bottom a lot of programs, free software that community has developed for everybody to use. They are maintained by people voluntarily. So, and it's a huge exercise so the science can be more open for everybody to use and to analyze and to share because maybe you work 2 years to be able to create a pipeline that works to segment an embryo and track the cells. Why not to share that so everybody can? You will have the citation as a prize for you to doing that hard work. I'm really scared to show you this picture. I really like it, but I think it looks really scary. So if we talk about data analysis just in this little corner, so the development of analysis tool, in this case, I'm talking a lot about the open source. That is quite a lot. And there's people that they will do the image analysis services for you. But this is just the little corner here for what we call the FAIR bioimage data ecosystem. This is a really beautiful dream that hopefully we will manage to go there. So you will have the data management properly. We will be able to open an image from one of the bioarchive or archive of bioimage and know what is in there, not only see, oh, it looks like a cell, but knowing what cell was staining with, the experiment, the drugs, and everything, and you will be able to either test your pipelines, that's what I was talking about, or even do run another experiment or another analysis and have more data coming from something that it was acquired 5 years ago. So that was also quite a really good impulse. This is go into the reuse. So I know it's a really huge thing, and we all get really, really overwhelmed with all of this. So that's why I'm in the same point of everybody else every time I see this graph, because it looks like a lot, but if you think about it, to do all these things is not only the core facility, it needs to be the researchers, it needs to be the institutions, it needs to be a lot of people working together to be able to achieve that as a end goal point. So we all panicking around thinking that that's going to be impossible, but nothing is impossible. So it leads me to the last part of my talk. I like to present you guys is that we are all overwhelmed with data analysis, especially what I see in my course facility. We are all overwhelmed with data management, or even how you acquire the image, how you answer your questions of biology. So I think it's important to build communities. There is a lot of communities. This one I'm going to present is local to UCL. We are open if you want to give a talk in our place, but the end goal of our community is to be able to work together and have less problems and less let's say obstacles to make the research to be more efficient. So the BIG is the Bioimage Interest Group. I'm part of the committee, but it was created by two people that I put here. Is Julia, a postdoc, and Alessandro, a software engineer. They created the group in 2020 with the same ideas of trying to make science to work more efficiently, especially in the context of bioimage for both acquisition and analysis. So the way we work is trying to put everything together. Right now, UCL based, even UCL based, like in the same university, is difficult because technicians we are tend to work very local based, so you don't know what is happening in the other faculty. When you are a PhD and a postdoc, it's even worse because you know the people in your lab, but you have maybe you are not aware that someone else is doing something similar in other department that will, even if you collaborate, your paper will be published faster, or you will have a better outcome of your experiment. So our idea is to try to put everybody together and try to work together to share our knowledge, to help with problems, and also to promote best practice, because we are very passionate about proper microscopy and data analysis. We are focusing, as I say, in students and early research, early career researchers, and also staff, technical staff, just because we think we are the seed for change. Normally, they start from the bottom, it goes to the top, these kind of things. And the way we work is, well, it's kind of similar to a virtual path. We have only a monthly event every, I think it's the second Friday every month. And we aim to bring two speakers. What I mean by different backgrounds is we tend to put someone that is doing something very fancy with a microscope, or is trying to do something very fancy with a microscope. And the second speaker, it will be someone trying to do something with data analysis. We also welcome people about other communities, and also from companies if they have something interesting to share. It's really informal, so literally you can just put your presentation that this is my problem and nothing works, and you have several slides of this doesn't work, please help me. That's actually quite useful because you will have like a chamber of people from different backgrounds giving you a lot of ideas and to work with. We offer food if you are local to London, and because it's a hybrid kind of event, you can join online from everywhere and listen to our talks. But if you're lucky and you can come to London, we normally discuss after the talk with pizza, and it's free. So that's also for the early career researcher, it's a good thing. We also organize summer events. We normally have a hack day. I'm going to explain what is that, because the first time I saw that, I thought it was something like very cool, like hacking stuff. It's a day where you spend with different projects, working with different people, coding to solve a problem. Even if you're not an expert in coding, like I was the first time I went to the hack day with these people, you learn a lot because you will see how people solve a problem in data analysis through coding. We also offer training opportunities. Actually, the introduction to image analysis is organized by one of our committees too. It's here, Kimberly. And we will be also organizing an imaging symposium here in UCL, because even though I work in a core facility, I don't know how many microscopes we have in UCL and who is doing what. So that's how difficult it is to build a community, but we are working with that. We also have a Slack channel to people just to also literally, I don't know how to install my environment, or I need this white field, where is someone can find something with this, I have this fluorophores, there's any microscope I can use. So this is another QR code in case you want to sign for our newsletter. I think if you are not in UCL, it's difficult, but you can also go to the LinkedIn web and you can follow us because I tend to put everything in there. And get in touch if you want to give a talk. If you have London in your plans to visit, it's always a good excuse to give a talk. We cannot pay anything, sorry for that, only the pizza, but we will. We can spend a very nice showing you all the microscope here. And just as a final point, I put this image just to show you that if you, in case you are curious about what is the technical career, it's more or less that this Swiss knife. I don't even know what these things does, but it's also what happens with technicians. They will come to you and say, I have this problem, and your goal is to help as much as possible for people to do their research as fair and as good as we can do. And I think I finished because I'm very quick. I put here all the QR codes just in case someone else wants to check them. And happy to take any question if you have.